Welcome everyone, and thanks for joining this webinar. I'm Arne, founder and CEO of AcademicLabs, and today we'll focus on how scientific scouting is changing and what that means in practice for teams like yours. Most scientific scouting teams don't have a data problem. They have a signal problem at scale. Today, we're dealing with thousands of labs and startups, millions of researchers, and every year, tens of thousands of new projects and clinical trials. And on top of that, millions of new patents and publications are added continuously. Challenge isn't finding information or accessing information. The challenge is finding the few signals that actually matter and doing it fast enough to influence real decisions. In this session, I'll show you how teams in pharma, biotech, or investors and consulting cut through that noise, rank entire scientific landscapes in minutes, not weeks, using real evidence and not gut feel. This won't be a live walkthrough. I've pre-recorded each use case and removed all the waiting time. So I'm going to move fast. There's no need to memorize clicks or write down the prompts that I'm using, because after this session, I will send you a workflow guidance document with every prompt, search query, and filter setting I used today, as well as the outcomes. So for now, you can just watch the workflows and think, where would this help my team find signal faster and make better decisions? So I've structured this session around three types of decisions teams struggle with the most. First, decisions about people and partners. Who should we talk to? Second, decisions about technologies and IP. What innovation actually matters? What is relevant to us? And third, decisions about diseases. Where to focus? What signals really matter? Its targets and novel biomarkers and trial strategy. Each section builds on the previous one, and together they show you how the same approach can be applied across very different scouting questions. Any disease, any technology, any targets, anything of interest, you can apply the approaches that I will demonstrate to get to the desired outcomes quick. We'll start with people and partners. That's where historically AcademicLabs has been the preferred solution by many. Now with the new AI-powered capabilities we launched, our customers have been extremely excited. So I'm excited as well to show you what you can easily achieve yourself. So this use case answers the question, who are the experts we should engage with and why? Let me show you how teams evaluate experts based on evidence, not just reputation, and with scoring per criterion of interest. When most teams start scouting the search for experts, instead of browsing literature or event keynote speakers, with AcademicLabs, it just takes a few clicks to find any expert on any topic. Let's say we search for atopic dermatitis, we find immediately 92,000 researchers on a topic. For example, if we search for academic researchers who had a recent publication in the last year on a topic, plus a trial in the last 10 years on atopic dermatitis, in just a few clicks find these 140 researchers. So now the bottleneck is no longer finding these names, about deciding who actually matters most for your strategy. And because a top KOL doesn't exist in a vacuum, the best expert for you depends on what you need right now. Is it disease biology knowledge, discovery insights, technology strength, translational depth or trial execution, patient access, or even commercialization? So traditional scouting favors rather visibility that allows voices, keynote speakers, scientists everyone recognizes, the authors with the highest age index, etc. The real competitive advantage sits with the people who are producing the most relevant signal right now, in publications, in trials, in grants, in patents and translational work. And sometimes those aren't the obvious names. For example, if I click on a researcher here, who is associated with the hospital and with the universities and as well with the new Pharma, we have immediately a tailored summary on atopic dermatitis. So we see what's the main focus of this researcher with recent signals, what this researcher has been focusing on. I want to see if Jacob is a perfect fit for our KOL needs. Once you define what ideal fit means for you, for example, deep biology expertise on the disease, drug discovery, focus, experience with translational work, strong reputation, industry collaborations, some other criteria, and why we need a KOL, can save it, can make it a default analysis, then search for atopic dermatitis within this researcher's research. I hit evaluate, and now the assessment is excellent. There's an instant assessment on her criteria with a score on 10, with always justification and reference to, for example, publication of 2025 in that topic. So we get an instant assessment to see how well this researcher fits her criteria plus some other criteria. So this is not for one KOL. The real value is in analyzing many KOLs to see which one fit best and why. And all that takes is a few clicks. So if I go back to the search, we have 140. I go for the AI and I evaluate all 5,000 researchers, but up to 50 with a report. So I select 50 KOL fits. I still have this criteria available here because I saved it in my previous step. I want to analyze the 30 most recent publication atopic dermatitis of each researcher, the 30 most recent trials, the 30 most recent patents and grants. So for each researcher, we will look at the recent volume of work on the disease. And then I can define what I want. The goal of this report is to identify the best fit KOLs. I want scoring on different criteria, in-depth assessment of the best fits candidates. I hit evaluate, and now all these KOLs will be analyzed. Then I click on view report, and I get my summary. I get an introduction on the topic, scoring for different criteria, and then I have a comparative table. So this allows me now for each KOL to identify for the different criteria of interest to me how well this person scores with a data score, a KOL fit assessment, and a brief justification. With links, but we also get an Excel file with the full detailed description. We see some additional legends, so you know what you're looking at. You see different tiers, strategic innovation leaders, high-value experienced experts with a rationale, an example profile, and then more niche KOLs. So depending on your needs, you can identify now which ones make most sense for you. Here we have some extra information for top KOLs from tier 1. We also identify specific trends and patterns and clusters across those, for example, niche KOLs in pediatrics, microbiome, some opportunities at suggesting, challenges in engaging with them, and then finally some recommendations that we asked for. So all of this is just a few clicks to get this kind of analysis. You wait a few minutes to have the report ready. And if you want to go deeper into the evaluation of a single KOL, you get the expert file with the 50 researchers here. For each researcher, we have an assessment. So we see overall fit excellent, and then we see the different criteria with a score, and then justification like our recent multicenter phase 3, etc. So with such an assessment of each researcher, you can easily identify which ones are the best fit for you. You can simply as well filter for the ones, say, excellent, and in this way, get 34 of the 50 that score excellent, and then you can deep dive on the ones that make most sense. Now it's in this Excel file. This will be built into our platform in the coming months so that you can, in one environment, do all this analysis, and you can always export things, of course, to Excel. So what does this now mean for you? This turns scouting from collecting names into making decisions. Instead of manually reviewing 5, 10, 20 KOLs and missing 50, 80, 100, you can now evaluate 50, 100, 200, even up to 2,000 experts on any topic to know exactly which ones you'd like to engage first. So you basically go from debating lists to aligning around evidence, which removes the politics and accelerates the decisions. So you don't replace judgment. You focus it where it matters most. And the real ROI isn't a list of experts, it's the clarity and who you should call tomorrow and why. All right, let's move on to the next use case. Now, where KOLs help answer who shapes thinking today, rising stars help answer who will shape it next. And that's a very different scouting question because these people are not yet on the conference keynotes or advisory boards. Once someone is already a household name in the field, everyone already knows them, and access becomes slower, a lot pressure, or too late. So here, instead of starting with reputation, we start with signal velocity. So if I search for atopic dermatitis, we want to find the academic researchers who are currently active on the topic. So who published in this year, past year, on atopic dermatitis. And we want people who have momentum. So we can see here that every year they publish and it's increasing. We also want people who publish in top journals, atopic dermatitis. So who published in Nature, Science, Cell, Lancet, and so on. So we can simply select top 20 journals. And then we want rising stars, meaning we want those with probably around 10 to 50 publications. And to have a lot of citations on the topic, but that could be just a single publication. So the H-index in that regard could be a helpful way. So an H-index of 11 to 50 while having about 10 to 50 publications. So that's a very interesting comparison. Now, for any rising star, we get a tailored summary of their recent research on the topic. You see immediately that's the focus of this research. We can see the recent signals. But what matters truly is whether they're growing in the direction of your needs. A rising star in target discovery might not be the right fit for someone needing trial executions. So again, it comes back to contextual fit. So if we check rising star fit, we can see, okay, what does an ideal rising star look like? There's some defaults here. You can add something specific, expertise in atopic dermatitis, focus on drug discovery, clear differentiated niche. And let's add our scouting context. We want this at default. I want to save this analysis and search in atopic dermatitis in this profile. So in seconds, you go from guessing based on publication counts to a grounded understanding of whether someone is emerging in your direction. And critically, you see why. So it's an excellent fit, and you can see the different criteria with a score 10 out of 10, 8 of 10, plus the context. Now, where this becomes truly transformational is when you scale. I can evaluate up to 50. I go for the rising star fit. The goal is to identify high-quality rising star collaboration opportunities, and we can hit evaluate. So we gather a report after a few minutes, and in such a report, we can identify who are the best rising stars for our criteria. So we see a table, we see the different criteria, and the scoring on each of those criteria with an overall score, justification, etc. So we can pick the ones that fit our criteria best. We can also look into the in-depth assessment of different categories of rising stars. We can as well see which people score more than 58, but as well identify patterns across all those specific researchers. So we can see the different clusters of researchers. So it makes sense potentially to work with one of those or a combination of those. So we have some recommendations and a conclusion and the legend, of course, for the table. Once we identified the rising stars we want to assess in detail, we can simply go to the Excel file with the individual analysis of each of those rising stars. So we can see basically for each researcher, what's the overall score? And let's say we pick the ones that have an excellent fit. So we filter excellent. And now we can go to the ones that are having an excellent fit. And we can see the scoring on each individual criterion plus the context. So we can basically in this way assess any researcher for the rising star fit based on the criteria of interest to us. So that's a very quick and easy way to analyze 30. It could be even done for 2,000 researchers that you can assess how well of a fit they are for your criteria to basically map out the entire world for who you want to consult or collaborate with. So this turns rising star scouting from being a side quest into a systematic approach to build early relationships with the next generation leaders before your competitors are doing this. And just like with KOLs, the point isn't to replace your judgment. It's to focus your attention where your future advantage is. When you see who's rising today, you gain access before the rest of the field notices. So we've covered who shapes thinking today and who will shape it tomorrow. Let's now move on to the next use case. This use case answers the question, which research groups are the strongest collaboration candidates? Here the focus is collaboration fit, not just scientific reputation of a research group. Let's get started. So how to find ideal collaborators? By the way, this is the page that you will receive where we have all these different use cases and where you can see what are the steps to be taken. So we're going to go through this very quickly now. So I search for mRNA delivery or RNA delivery. I find these research groups. I click on a group and I click on collaboration potential. Here I can list my criteria of interest. Let's say I have 3, 5 criteria. That could be an approach, But we could also list a more extensive explanation of, for example, let's say Novartis. They have certain expertise on this topic, but they also have some gaps related to developing better technology. So I got to this data by analyzing the Novartis profile in AcademicLabs to figure out their gaps. So if I just put this here, I can now assess the collaboration potential. And I save it. I let the AI come up with a query based on what Novartis has as gaps in this research. It comes up with this query, and now we hit evaluate. We have an excellent collaboration fit score. So we have a score per criterion of interest with an explanation and information about the source, addressing the Beyond Liver challenge central to Novartis goals, etc. So each of those criteria now gets a score and an assessment. So we have a very good idea. Plus, it comes up with ideas on how to position a collaboration in a smart way, how to combine Novartis' interest with this group's capabilities, plus how to communicate to this group leader in a smart way to get this person interested. And then you have a final summary. You can then get the email address to actually reach out directly. And if you want to scale this, you already saw in the previous videos, you can just with the AI do this for 10,000 groups, or 2,000 in your case, or 50 with a report. And once you do that, it looks something like this. 519 research groups which I assessed on mRNA delivery, and then we have for 519 groups all these scoring with then the detailed analysis that you can review here. So 519 research groups is of course a lot, so I want to figure out how can I prioritize them. You could already filter by excellence, but I want to have a faster way. So what I did is I uploaded this Excel file into ChatGPT. And I said, where is the data? I want to know, I said, these are the gaps we have at Novartis. And I want to know what are the main complementary archetypes of collaborators? How do they differentiate? And within those, what would be the best groups to collaborate with and why? So then after 7 minutes of analyzing the data, it comes up with 47 groups score excellent. And of those, these are the 6 archetypes. So different types of expertise with then some extra information, summary. Per archetype, it shows me the best candidates, the top 3, how they differentiate, the confidence about this analysis, and then the other archetypes, etc. So for each archetype, I know exactly who I should best focus on. And then a short list of the key labs here, the top 8. Then some trends across all those, challenges, recommendations, etc. So this is something where you could assess hundreds or even thousands of research group collaborators or startup collaborators and have a first assessment and then run it into ChatGPT, for example, to extract exactly what you seek. And that's, by the way, something we will integrate within the platform, all these steps so that you don't have to take these different steps. Copilot is not great in doing this, Microsoft Copilot, compared to ChatGPT. So we will build this into the platform so that eventually you can do this and then you can ask follow-up questions, etc. Right, so I hope this helps, and let's move on to the next use case. This use case answers the question, what is the technology behind the startups and how strong is the technology? We'll be evaluating each startup individually, not forcing comparisons per se, although that is an option. So we look at the validation, the stage and differentiation of the technology of those companies or those startups. So here we're going to start from pharma companies' partnering interests and then use that to see which startups have technologies that match those interests. So let's say at Pfizer, they're interested in bispecifics. I don't have much more detail. Of course, if you work at Pfizer, you know a lot more details, criteria, etc. you're interested in. So what I will do now as an example is I go to the Pfizer profile, I ask anything, and here I ask, based on the developments with regards to novel specifics, summarize the innovation interests, and call us to use this information to compare any new technology with, to see how well it may fit Pfizer's interests. So then the AI comes up with a query, and then we execute that query across the 160 web pages, the recent publications, the recent projects, patents, trials, news articles, to identify what is Pfizer interested in related to multi-specifics. And then we quickly see here, okay, in oncology and hematology, what they have, what they're interested in related to manufacturing innovations. You see clinical focus based on their clinical, preclinical IP activities, strategic teams and external innovation. So a summary for fit assessments. So I copy this. The next step is now to find companies. So I did a query here for multi-specific or bispecific or tri-specific. So it's 3,500 companies. That's all. You can see why they match, because there's 470 patents, 125 publications, they're mentioned on the web page, etc. Now I can, for example, focus on the preclinical therapeutic startups. So let's say I pick the first one here. It will automatically write a summary on what this company do on multispecifics. What are they doing exactly? What are the recent signals of their activities in this context, with always a reference to the source and exactly the sentence that will be used, plus it's from 2025, and what was the title of the publication. So we see what they do on this topic, we see the recent signals and why it matters. It's a front runner. I could do additional analysis. So I could, for example, look at the technology, the key technology. So I can, again, search for multispecifics across the research of biomunics. It will then assess all of that and come up with a one-line summary, and then the key technologies that they have available. We can see application examples. We can see technical validation, strong preclinical validation, and relevant models, including high throughput in vitro cytotoxicity and organoids, patent portfolio across many different targets. We see IP and enabling tools and key insights. So in this way, the one click, I can assess the key technologies of this company in the context of multispecifics. An alternative approach is looking at preclinical activities. So here it talks then more about the specific assays and studies they have done, the outcomes, etc. We see the disease models, we see the functional mechanism studies through the key insights summary. So that's how you can assess specifically the preclinical research. Now I could also check the drug research. So here I just say multi-specifics, evaluate, and now assess what's in the pipeline with modality, the indication, the MOA and the targets, the stage, preclinical, scientific validation. So all of that, so you don't have to search, you don't have to click around. It just gives us information readily available for you. You see as well, boundaries they have, IP. So we see it for the different assets that they have in development, and then the key insights. So now we're also building a full asset intelligence solution where we extract all this, we structure this across any asset, across any therapeutic area, from very early stage data to late stage data. So that's something we're working on and will be released in the coming weeks. So the last thing I want to show, very powerful one, is analyzing opportunity assessment. So let's say we're interested in bispecifics or multispecifics, and below I add what we had on Pfizer. So I have all this information on Pfizer's interest based on their scientific activities. I save this input, I use it as my default analysis, and I can now run a query on, let the AI come up with a query based on all the things we're interested in. So evaluate, now it will assess the technologies, the assets of this company based on what we provided. And here it says maybe, not an excellent fit, but the explanation then, interesting target for further due diligence. We see the promising assets, we have a platform, we have explanation, the stage, the validation scientifically, mechanism of action. We see then different assets, preclinical, in vivo validated state, also preclinical state here. Now, interestingly, this is not for one company. I could do it for up to 1,000 companies. So all 818 companies I could assess with the AI. And I click on AI, and then I evaluate up to 1,000. I won't do all of them now, but you can do that. It takes about 5 minutes. I'll choose the first option where we can also get a report. So that's even more interesting. So let's say we do 50, we check in-licensing opportunity. It's still here, advanced settings. We will just search for anything bispecific, multi or tri-specific, and any company profile. So what it will do, once we hit evaluate, it will assess the 30 most recent publications, all these multi-specifics, the 30 most recent matching patents, and the same for grants, clinical trials, news, and the document pages. So in this way, it has a lot of information available to assess how well of a fit this is. Then in the report, we could say identify high quality in-licensing opportunities. So let's hit evaluate. And now each of those profiles, the first 50 will be analyzed in about 2-3 minutes. the report is ready, if I click to view the report, I can download the Excel file with the evaluations of each individual company. We see an executive summary, emerging trends, impact industry leading candidates, and so on. If we go down, we can see as well a comparative ranking of companies where we see different criteria, the score out of 10, the total score, and justification. What differentiating between the tier 1 candidates, which are late preclinical to clinical stage. We also see alignment with vice versa fit, then the earlier stage of portion fits candidates. So comparing preclinical data, some key differentiators are listed here, and then more early stage technology adjacent or non-oncology first companies. We see why they differentiate, and then in those tier 1, you can see key companies with specific context on those, an in-depth assessment of the tier 1, the best fit opportunities with scientific validation, the stage, the summary, and the assets basically. We have more information about the hotspots or trends, rise of tri-specifics and multi-arm TCEs, for example, protein degraders, plug and play, etc. We also see some clusters around indications, IP hotspots. Comes up with as well challenges with target toxicity, for example, freedom to operate, cytokine release syndrome, key opportunities, and some watch outs and then recommendation. So that's what we get in the report. In just a few minutes, you have this total assessment of 50 companies on the specifics. Here, we're going to look into the individual companies. So the 50 companies, they were analyzed. We see here the different companies, and we can just look at where it says yes on is the good in-licensing opportunity. We can see why, and we can quickly move to the different ones. And we always see the context, the most promising assets, information about the validation, modality, IP, partnerships, etc. So we can easily scroll through the different ones to do a deep dive. And you can always learn more about these companies, where they founded, least funding rounds, the specific TAs and sub-TAs they focus on, specific modalities and sub-modalities. And you can go to the profile on AcademicLabs, the website, to review them more in depth. So that's how you can easily go from any company on what the specifics. You process up to 1,000 of them. You have this kind of assessment, or you scale up in smaller chunks and analyze up to 50 with them per report. And that all in just a few clicks. So I hope this helps. Let's move on now to the next use case. This use case answers the question, what has changed recently in the startups you're tracking? This is especially useful when you're monitoring dozens or even hundreds of companies over time and you want to figure out what's changed, what the latest scientific advances are. So you get a per company progress assessment. So let's say you have companies you want to track, whether from an event, from companies you have been tracking or speaking with in the past months or even years, so that you want to have a regular update on. So how can you do that? Well, let's say we have a list in Excel with a list of company names. Can be dozens, could be hundreds, or even thousands. Then we have a simple formula I'll be sharing with you to add quotes around them and then make a search string. So what I do is simply copy the search string with all these company names between quotes. I search by company name, and then I search for this list of names, and it goes very fast. So it was 50 companies, here it says 64. Why? Because sometimes there's probably a company that includes maybe also biomunics or some other term. Now it's not at the end of the world. Why? With the AI, we can process the data anyway, and we will only retain the ones that are relevant. So how do we go about this? We simply click AI, we evaluate up to 1,000 companies, we select all of them, and then we ask anything. When we ask anything, we ask multiple questions like, what is the most advanced asset? Mention name, indication, MOA, and modality. What's the latest scientific validation of this asset? Question 3, what's the key progress of this company in 2025? Or what are the negative events for this company in 2025? We could just let it do an empty search without anything. That's something I would recommend. So I just hit evaluate, and now each of those profiles will be analyzed based on the most recent documents to figure out what's the latest progress. Now already 20 are being added and we're just 10 seconds for. So it goes pretty fast to analyze all these companies. All right, so about 30 seconds later, we were able to get the results. I downloaded the Excel file, and now I have these 64 companies. So let's look at the first company here. You see the most advanced assets. So it's ISB 2001 and so on. Was the latest scientific validation of this asset. Received scientific validation, promising clinical trial results in 2025 presented at ASCO. So we see specific results, and it comes from a news source and another news article. Now, was the key progress of this company in 2025? Pivotal progress for IGI in 2025 includes the global licensing agreement with AbbVie, receiving this kind of payment, and as well, interesting results. Both are negative events. Here you can see no reflect robust clinical progress and no setbacks. So for each company here, you could in this way easily see what is new on their assets and key progress in the year. How do we do this? We analyze the 30 most recent projects, the 30 most recent patents, the 30 most recent publications, 30 most recent news articles, top 30 web pages, and clinical trials are the most recent ones, to analyze any changes based on the questions we have. So in this way, if you do that every 6 months, every year, you could assess a whole bunch of companies to see what's changed and maybe something to follow up with some of those startups before your peers are done. So all in all, it's a very convenient way to analyze all these data points for every company in just a few clicks and a few seconds of work. Hope this helps. Let's move on to the next use case. Next, we move from people to technologies, where the challenge is separating real innovation from volume noise hype. So this use case answers the question, where is novelty emerging for any technology? So for this use case, we'll take a different approach. We'll landscape new technology developments in a particular topic. So let's say we focus on antibody drug conjugates published in the last year. With AI, I can extract insights from all these patents. I can just select all of them. For going fast, I just select now, in this case, 200. I let AI add 20 questions. I want to landscape any novel ADC innovations to identify opportunities to license and so on, and assess the competitive landscape. Now the AI comes up with 20 questions. So now we get in one sentence, what's the most innovative part about it? We can see with the multiple choice questions, what is the primary novelty type, antibody, payload, linker, conjugation method, and so on. And if it's other, it needs to specify. So we also pick up a new kind of novelties in this way. Same with indication, the payload class, with kind of payload class, we'd always have to come up with 10 to 20 options, and then for other, always specify. The linker strategy, the validation, organization involved, and so on. So it pulls out a lot of interesting insights. You can adjust these questions to your liking. If I then click show preview, I get the first 5 analyzed, so I can adjust them. And if I'm happy with that, I can click on download, and then I get an Excel file like this one. So I've run the analysis before. We have the 20 questions, the query, and then we have our AI answers. So summary of the claims in just one sentence. We see what's the primary novelty, the primary novelty type. So I can say novel payloads. I can just select those. And now we have 39 of the 200 patents that are about the novel payloads. So you can see, yes, it's a first-in-class ADC novelty. It's about this radiometal. We see development stage, the positioning, which kind of organization. So I can pull out any biotechs, and then I see 13 are about biotechs, and I can see which ones. So the target antigen, the conjugation linkers, it's the new class ADC. So yeah, a lot of interesting insights that you can just pick and choose the exact patterns that are of interest to you by selecting the right questions and the data points you extract. So this is a very easy way to scrutinize any new patents. So I could do this for the last 3 years and 5,000 patents on this topic. I could just filter by the type of novelties and see anything that is newly patented. You can do the same with publications and so on. And now I can even throw this now into ChatGPT and ask to assess the landscape to identify these kind of opportunities. It says what the innovations have been skewed towards. You can see the summaries. This amount of patents are focused on this, etc. So innovation is mostly in antibody and targets. Here we see the exact numbers as well for solid tumors versus broader oncology and non-cancer indications, innovation archetypes, shortest of opportunities, and then I can ask follow-up questions, because now we have this structured data set, so we can ask many questions about this. Then it thinks for 5 minutes, analyzing the data, and then it pulls out the top biotech companies, and then why as well, why do they stand out? So in that way, you first search for all the relevant patents or publications or trials or news or grants. You come up with the right question, the AI does that for you. You can then select the right data in the Excel sheets. You can then filter data for the ones that are relevant to you. You can assess it in this way in Excel and then upload it into an LLM like ChatGPT to ask any kind of question. And that's something we will build into the platform, of course, soon, so that you can have the full experience within one place and to make it as fast and easy as possible, of course. All right, hope this helps. Let's move on to the next use case. You don't need to be an expert to do a pre-screening for freedom to operate analysis. A lot of innovation, search and evaluation teams, intelligence teams, R&D teams, they can help their IP attorneys to already pre-screen a bunch of patents that could have an impact on their freedom to operate, to then only share the ones that are really relevant with the IP or patent attorney to then do a deep dive analysis. How to do that? That's what I'm going to show here in a few minutes. So if I search for mRNA delivery and lipid nanoparticles, we can simply analyze all these patents. With the second option, extract insights from individual patents. It will analyze the abstracts, the titles, the claims of all the patents, and generate an Excel file with each individual patent that will be analyzed. And we can ask up to 20 questions for each. I won't do all of them for this demo. You could easily do all 810. Let's say we do 400. I can ask to 20 questions to extract specific findings, so we could filter out irrelevant patents as part of this pre-screening, and then only retrieve the relevant patents to share with our attorney. So how do I come up with questions? I could individually come up with questions, but it's easier if you just let the AI get 20 questions. So I describe what I want, pre-screening, with regards to novel NMP formulation, we are considering why. Let AI add 20 questions, and the AI comes up with a few key questions. So first we have a claim summary, a few sentences. We have an idea of one sentence was a primary inventive concept or claim method. So if I now click download, it analyzes these 400 patents in about 1 or 2. So what does it look like? These are the 400 patents. We have the 20 questions here, and then the query, etc. to repeat it. So this can be shared with the IP attorney to just repeat the same on AcademicLabs and see what was the steps to get there. What is very important with AcademicLabs is you don't need to do like a lot of searches combinations. You can first come up with the broader query that captures everything and use the AI then to find the specific patents. You hear from IP attorneys that otherwise they have to do a bunch of different queries to combine data sets of patents to make sure they don't miss out. But then it requires a lot of screening through all the noise. In this case, I could say, okay, only where it's related to mRNA. Well, we have 352 of the 400. Only if it's about cationic, then we have 39 of the 400 records. So you can see in this way, I reduced 90% of the noise in just 2 clicks. I could see which are the parties here that are involved. So Sanofi seems to be the key party with these approaches. Let's say I'm interested more in regulated ones. Then we see some other parties. What's the LNP component? We see the specifics here. If I open up the filter, I can look into the details if that's something I'm interested in without having to read the full. We see its broad platform or specific use cases. We see what's the main challenge, what's the main risk. So we could look into the details of those to assess what could be a risk. Now, what we can also do is upload this file in ChatGPT, for example, to ask questions about it. So I've uploaded this patent data set. I mentioned, help me identify what's blocked, what's the white space, and which additional due diligence I should do. So here we see concise answer to your core question. Most likely blocked if your candidate LNP uses this one here. We see you appear to have white space if your formulation relies on this and that. And then some critical next step here. An executive summary, we can see what's available amongst all those patents, what is likely blocked, specific examples, why it blocks us related to this kind of classes. the disease limitation, and so on, where there's white space, what additional due diligence to do. So the interesting thing is it will now always use the structured data set that we extracted, and you can ask any kind of questions about. Of course, you can rerun an analysis with different questions to assess more in-depth claims for specific areas that you want to investigate. But in this way, you can really help your IP attorney in maybe 15 to 30 minutes to pre-screen relevant patents in space and reduce 90, maybe 95 or even 99% the noise so they can focus their efforts well. All right, I hope this helps. I'm happy to help out with these kind of approaches. Let's move on to the next use case. This use case answers the question, what first-in-class technologies are emerging in early development at startups and companies? So one approach to find novel innovations in early development is looking at funded projects. So we could say, let's look at funded projects of 2025, 2027, starting in any of those years. And we could look at academic projects, so more early stage, or we could look at where companies are involved. So I could say any company, but I think more interesting is the ones from biotech, pharma, tech, biodiagnostic, medical device, health tech, software, data vendors, research tool providers, and capital equipment. So we have 2,200 projects starting this year or next year on any topic. With the AI, we can now analyze each of those projects. So we just say analyze all 2,209, and we describe what we're looking for. So I say, I want to identify the most promising innovation in life sciences across these new projects with specific interest in first-in-class innovations or those with massive impacts. I want clear segmentation of innovations. Let AI add 20 questions. So we then get here the questions, what's the core innovation? Is it about drug discovery, biomarker development, gene therapy? Is the first-in-class innovation? So we have a bunch of questions. We can click show preview and then download the data sets. So we have here the questions, the 20 questions. We have the answers here. We have the project goals. What is the technology adoption relevant for pharma? We could say yes to those because we are a pharma company. So now 160 of the 2,087 are left. I'm interested in drug delivery. Is it about automation, genomics, etc.? I'm interested in early validation, but also concepts, prototype, proof of concepts. So the early stages. So 75 of the 2,000 projects are retained. I'm also interested in novelty to be first of kind or significant advancement. So here we can then see what's the competitive advantage for pharma, the unique features of the technology, and what's the breakthrough potential. So we see all these projects here, and then at the right we see the details of the funding and the parties involved. Now, in this way, you can quickly skim through a lot of interesting new innovations and select the criteria of interest to you. Now I selected drug delivery. I could easily adjust to diagnostics, drug discovery, etc. It becomes even more interesting if I upload this into ChatGPT. I asked, give me the hot technology classes with a quantitative distribution, the most promising first-in-kind or massive improvement technologies for the relevant tech classes, and other insights. So here it gives me distribution. There's a lot in omics and AI, of course, these days, imaging, automation, high throughput, etc. Then we see most are for drug development and drug discovery. We see for different classes what are some very interesting technologies that show novelty, breakthrough potential, validation, maturity, scalability, and pharma adoption. So it needs to be yes for those, and then it pulls out those technologies. So that literally took 5 minutes to get to this in just a few clicks. We see the same for genomics, advanced imaging, etc. If I further go down, we also see which parties provide most of this funding. You see how many are big projects of more than 2 million and smaller ones, which stage are these projects. Now, if I go down, I ask the more specific taxonomy for the ones that are mentioned other. So then I have digital and computational platforms, experimental discovery engines. So in this way, you could identify the types of technologies that are being developed. Here I asked what are the first of its kind or significant improvement drug delivery technologies being developed. So now it specifically filters through that data set that we created to pull out the three generally interesting delivery clusters. Smart micro robotic delivery sounds very cool. So we can see here what it's about, what's the maturity, targeted nanocarriers beyond classical LNPs, and examples, and then controlled release and biodistribution engineering with significant improvements. Formulation platforms, they're undervalued but strategic. So here again, some options always referring to specific cells or evidence. So that's how you could, in just a few seconds, analyze any new projects on a particular topic to see what's cooking, what's next. You could maybe engage with those parties to be involved, to co-develop, to make sure they go in the right direction, or that you can license them first before anyone else. All right, hope this helps, and let's move on to the next use case. Finally, we bring everything together at the disease level, because this is where signals translate into real strategic focus and investment decisions. Here you'll see different workflows depending on whether you're looking at late stage or early stage signals, novel biomarkers, or innovative trial designs. Common question we get is, can you help us find early stage assets that show promising signals? So to find early stage assets, it comes down first with how can we break it down into a good query. Often it includes three parts: the disease and mechanistic terms, and more action and modality terms. So you can use LLMs to come up with a bunch of keywords. That's something we will integrate in the platform too. So let's say we do the search in projects, and we filter down the projects by the ones that are starting to 25, 26, and we ask the AI a question about these top 50 projects. So we ask anything about the set of 50 and select the first 50. I ask the question, what are the novel early drugs and treatments in discovery development for AD? And let's list them in a table with per column interesting criteria. So literally in a few clicks, I see different new approaches being developed, the mechanism that they discussed, the modality or technology, the need for organization, and the stage. There's always a link to the project. So I have in just a few clicks an idea what new innovations, new projects are starting in the past year to find novel AD drugs or treatments. Now we could do exactly the same by going to ongoing research. So we use the AI and we ask the same question. So this is ongoing research, basically what research groups in academia communicate on their website that they focused on. So again, in a few clicks, we see new approaches being developed. So all of that just takes a few clicks and you already have a pretty good understanding of what is being focused on. Now let's look into publications. We got 3,700 pretty relevant publications probably, but not all of them are relevant. So then we use the AI to scrutinize the data. So with the AI, I can extract insights from all these individual publications. So I simply ask, I want to find very early asset opportunities for AD. And it now comes up with multiple questions. What's the novel therapeutic approach? What's the pathway? It can pick from options, plus if it's older, it needs to specify. What's the evidence? What's the modality? What is the disease biology? The organizations involved, and so on. So we have all these questions and simply need to click show preview and then download, and I get the full list. I've already done it before. So the questions we're asking is this about a drug in development? Yes, answer with the drug or asset name. Does it explicitly describe a pathway or target being impacted in the context of AD? So all of these we can now filter by. So I can see, is there a target or a pathway intervention in the context of AD? Yes, and then I have 257 of the 400. I can see here all the drugs that are being used or the assets. I can see also answers to any of the others. So I can see what's the role of the biological target and pathway focus. So I can say anything related to the ion channel. Now those are retained, say 13 of the 400. And we can see it's a central driver or contributing factor. Which kind of cell types are being studied? Small molecules, the highest experimental context. Is it in human, in vitro, human tissue, etc.? What kind of model? What kind of evidence is there? What kind of modality? What's the developmental maturity level? What's the efficacy results? You see what kind of organizations are involved. What kind of scope? Is it AD specific? So we could focus on things that are also broader applicable. And we see clinical relevance is discussed, etc. So we go from 400 publications to 3 that might be very relevant. So in this way, you skim through all the research on a particular topic and filter down by the criteria of interest. to find exactly the assets you might be most interested in. So another approach is by looking at research groups. So we could either look at research groups or any companies that have assets on this. Now, in this example, I'll use research groups for early assets. So who are the groups who have a publication in 2025 on the same query? So with AI, I can evaluate all the groups. And I'm going to do it just for 25. And I can ask anything I want to ask. 22 questions I can simply add, just because I want to extract a lot of insights on what this group is doing. So is this group developing a new drug for AD? Answer yes, no, or unclear. If yes, specify the name of the drug. And also target, what's the pathway? Is this a novel target or underexplored in atopic dermatitis? What human relevance evidence is provided? So we have a lot of different questions we can ask here. If I hit evaluate, now these 25 groups will be analyzed. But as you know, you can analyze all 377 groups who have at least one publication on this topic. And once we do that, you get something like this. You have these research groups. Each row is one group. And for each research group, we have the answer to each of those questions. So this one answers yes, this group is involved in this and this and that. We see a source for 2025. We see what's the target. It's an emerging target. It's on patient samples. It's disease modifying. Keratinocytes are being used. It's an antibody. It's in vitro and vivo and ex vivo human skin models, and so on. So you can see all this data, but just not having to search for it. You let the AI interrogate all the data we have about each research group to extract exactly what you see. And this way you can go through the different groups. So that's already a very powerful way. Now, what you could also do is take it one step further. We have now the publications, we have these research groups. We could throw this into ChatGPT to then ask questions about it. So that's what I did here. I uploaded both documents, both Excel sheets, and I said I want to find the most promising early stage drugs in development for AD with strong early validation. So it thought for 4 minutes, uses code to segment and interrogate the Excel sheets to come up with key early assets. It's always referring to the cell index. So it comes up with these 10, an executive summary, some specific questions, which early stage assets show the strongest early validation, and then it gives some specific context on each of those, what trends or patterns across those publication data sets, so we can see different patterns that it identifies, which research groups look most promising amongst those 25. So in this case, I didn't upload all the 300 research groups, but you can see most partnerable, translationally oriented. So you can see different groups here with each their strong signals of position or the context that is relevant. So it also says here how you can work with those. Now, then it gives again some short list of opportunities, some recommendations, and then I asked produce a ranked table with the top 15 assets, and then execute the 3 steps that was here mentioned. So here's then an asset table with the asset name, the pathway, modality, maturity, evidence, and so on, and then the audit trail you can use. It comes up with for these different assets, some specific data points it extracted from all the data that could be relevant for us. Then some questions to ask those parties when doing due diligence. So this is just an example of how you can use AcademicLabs in many different ways. You could find early assets by looking at new projects, at ongoing research, publications, even patents research groups. We could also assess preclinical startups or even pharma companies that really would communicate in an early stage. So this kind of approach basically allows you to pinpoint or find the needles in the haystack in just a few clicks. We are building out a full pipeline intelligence solution that will also extract all these signals from all these data sources that we have, including from websites and these pipeline diagrams from the websites of biotech and pharma companies, so that we can provide a full one-click solution to find early assets where you can easily filter and segment and so on. All right, hope this helps. Let me know if you have any questions. Happy to help you out in this. This use case answers the question, which late-stage assets matter in this disease and why? So here we focus on clinical trials, companies, and late-stage evidence. So while we're building out a full asset intelligence solution, we already have very powerful ways to quickly find interesting insights related to late-stage assets. So I'll show 3 ways. 1, we go via clinical study publication. So we have a quick filter, a search for atopic dermatitis, and a bunch of asset or drug-related terms for the last 2 years. And then with the AI, we extract insights from individual publications, we select all of them, and we let AI add 20 questions. I've already gone ahead with this, and these are then the questions it comes up with. Is this about the drug? If yes, mention the name, mention the dosage, and so on, the modalities, the mechanism of action, the results, the organizations involved. I downloaded this, and then I got this results. 612 publications with the questions asked, and then the answers. So I could see exactly all the drugs for atopic dermatitis in clinical studies, quickly assess them. I could see the treatment and the dosages, the therapeutic approach, the therapeutic class. So the AI came up with these options. And if it's other, it also needs to specify. So we never miss out on key information. We could filter by modality, by target, and this way we could see simply all the targets. If I all the mechanism of action, route of administration, by phase, so we could say anything in phase 3. So then we have 130 of the 612 publications or phase 3 studies. We can see the pharma companies, and then we can see here all the 91 of these phase 3 publications with the pharma companies involved, as well as who are these like hopefully working with specifically on those assets. We can see the population being studied, the novelty, clinical validation, scientific validation strength, the efficacy endpoints reported, same for safety. Is it first in class, best in class? So it doesn't take much time. You get all these insights that you can extract from clinical trials, exactly what you're interested in. So these are clinical study publications. We could do something similar in clinical trials. So we search only in the title, summary, condition, and interventions, interventional studies only from the last 2 years. And with the AI, we can analyze the individual clinical trials, we select all of them, and we let it come up with 20 questions. These are the questions it came up with, which we can, of course, adjust. I won't go through all the details, but it picks up interesting insights in those clinical trial records. And once we hit download, we got this data set from the 12 clinical trials with the questions asked and then the answers to those questions. So here we can filter by drug class, we can filter by phase, by intervention route, study arms to find specific insights on the different arms if it's with a pattern. Here, so we see what they compare with. You can see the primary endpoints, what's the planned treatment duration, organizations involved, age group, disease severity, specific variations of the disease conditions. We see other details that we can easily extract. And then, of course, you have the clinical trial. So all of that just takes a few clicks to extract these insights. Now, the last one, really the coolest one, is if we search for atopic dermatitis, how we filter for those with the clinical trial on this in the last 4 years. And with AI, we select up to 1,000 companies, we select all 186, and in Ask Anything, we ask a few interesting questions. So if I hit evaluate, it was coming up with the Excel file in just 3 minutes. So here are the 186 companies. The first one, CEO of Pharma, it's pretty active apparently on the topic. So you can see a short summary here. And then what does this company do? It's at the forefront of developing these advanced therapeutics. So we see different drugs in different stages with specific contexts, ongoing phase 3 trials and in children. What unique approach do they take? So as a specialist company in dermatology, you can see what exactly are they doing, what targets do they focus on, what did they patent in AD context. Recent patents include which experts do they collaborate with in academia. So we can see the collaborators with industry and key named experts in the recent projects. And then a conclusion. So we have that for each of those, for Eli Lilly, for Sanofi, Regeneron, Nestle, etc. So we can now have this analysis for each of those companies, what they do on the topic. Which is kind of a 360-degree view that otherwise you can't easily get from the pipeline database. Now, what you can then do is combine these data sets, the publications, the data we extracted, the clinical trials, and then these company assessments. So I uploaded the 3 files into ChatGPT, And I asked to landscape the clinical stage assets. And after 11 minutes of analyzing it, it gave me insights on 2 crowded classes, the JAK inhibitors and the 2 cytokine axons. Then some other ones, the OX40, with an executive summary on this analysis. You can see it's heavily skewed into biologics and small molecules. Then what's the stage distribution, what are the key drugs and which phases, etc. So we get kind of a quick snapshot of who is active, what are the approaches. So this way I didn't have to do any analysis, I just applied the files. Then it has an overview of key assets here, trends, and I could ask any kind of questions about this data set. I could ask specifics about the efficacy of the different drugs. I could have information about the key companies that are in that list. So the idea is you create a structured data set via clinical publications, clinical trials, companies active on the topic with the clinical trials, and then with this specific data point, you can ask any follow-up questions to interrogate the data sets to eventually map out the competitive landscape, identify opportunities for partnering, etc. All right, hope this helped. Let's move on to the next use case. In this use case, we'll focus on which novel targets show early but credible validation. So here we're going to look into how to find novel targets for a particular disease. Let's say we search for atopic dermatitis and novel target or new target. We see 671 publications, and let's focus first on the ones in 2020. So 89, I click the AI. I ask a question about the first 50 publications, so I select the first 50. I ask what are emerging new targets, what's the validation so far, and we get our answer immediately. So here we see there's novel immune modulators, there's this pathway, the OX40, there's JAK inhibitors, and so on. So they're emerging beyond the traditional Th2 cytokines. So we see novel targets emerging beyond the traditional ones, novel immune modulators, cell barrier pathways, alarmins, DAMPs, and others, always with specific information confirmed across this and that, with always the link to the source. And then at the end, we have a table with preclinical evidence, clinical evidence, and the mechanism, and a conclusion. So that's already the first way to get pretty up to speed. Now we can take it further. Let's say we want to scrutinize all the recent literature. Then we could apply a different approach. Say atopic dermatitis target, and we search only in the title and the abstract. So not in the keywords. And let's say we also pick 24 until 26. So now we have 1,600 publications mentioning target and atopic dermatitis. So now I could use the AI. I analyze insights from the individual publications. I select all of them. And here we add what we're interested in. So I say I want to identify for AD validated emerging and novel targets with clarity on the validation, understanding of biology, context, assets in development, mechanism of action, companies involved, etc. And then the AI produces all kinds of questions, the different validation stages of the target it has to pick from. We have here the targets, the novelty for AD. So new proposed target, we see what's a primary MOA. So we give some options plus other, and it needs to specify which parties are involved, what's the asset name, what kind of modality is it, patient populations, experimental validation. So a lot of interesting questions that we can adjust if we want. So I adjusted just the first question here. Is it about the target in the context of atopic dermatitis? Answer yes, then specify the target name. No, or unclear. If we do that, we update the preview. We can immediately see what the target is. So here we can see no, we can see yes. So if we click then download, in 1 minute it's been analyzed. And then we have these 1,600 publications with the 20 questions we have asked and the search query, and so I'll do repeated. First one, very important. Here we can see all the targets. So if we say yes, it will also ask always for the target name. So we see all of them here. So we have a quick understanding of all the targets that are being used in this disease. So we could just filter by yes, or we could filter by any specific one. P studs 6. Then we have maybe 1. It's preclinical emerging early evidence, signaling pathway blockade. We see summary of the findings, So we see here specifically what was found. The pathway targeting disrupts the central signaling nodes, and we see it's bioinformatics or omics. So in just one click or a few clicks, we identified a potential new target, or we can identify any targets being matched here. So then we can say, okay, I want to see if there's not like a clinically validated one, because then we might be too late. But let's say the emerging ones. So emerging early evidence. So let's pick those. And then we can see what's the validation stage. So then we could say anything related to preclinical. So any preclinical evidence of this target. So here we see then the validation and the biological relevance. So we immediately can analyze these 134 of the 1,600 preclinically validated emerging targets. We can see the targets and we can see the evidence here. We see the pathway, we can see the organizations that are involved. So I could say who are the biotech companies or who are the pharma companies. I can quickly see them here. I could filter by them. What's a therapeutic asset? So if there's an asset, we could say yes. And then we see all the assets, all the 83 publications with assets being mentioned. We see the type of molecule. It's a small molecule, agent, microbiome, monoclonal antibody, and so on. What's the model? Is it an animal? Is it in vitro experimental validation? The rationale is for the target in AD, and some other insights that could be very helpful. So all of that just took 2 minutes to run to extract all these answers. But now you have this very curated data set to figure out which targets could be relevant to take on in a new program, for example. You can always go to the right to go to the publication, and then from the publication you can go to the researcher to then have the tailored email being written to see, okay, how could we maybe reach out to that academic researcher that the conversation started. Now, what is also interesting is I could upload this into ChatGPT. So I upload the publication sheet, I said what it includes, and I want you to summarize established versus emerging targets, identify novel ones, list which ones have validation in preclinical or early clinical phase with context in the validation, and a short list of targets which should explore experimentally. So now the AI is writing codes to analyze the full data set, applying filters, etc. So very interestingly, after 13 minutes of thinking and analyzing the sheet that we uploaded, it came up with established AD targets, which kind of axis it is. So we see the different targets here. We see the most actionable emerging novel signals for targets, and then which ones are hot but come with a clear risk flag because programs are terminated or there's lack of efficacy. So we see here an executive summary. What are the key target axes, best experimental shortlist, and then we see here a breakdown of the established or clinically validated targets in AD. So we have the drugs, and we see always a reference to the specific cells of the analysis, the emerging novel targets, so IL-18, clinical proof of concepts, preclinical rationale in AD-like inflammation, novel mechanistic angle can be interesting. So that could be very interesting. We see the OX40 targets, IL-33, IL-1 beta, and so on. So here we have done an overview of the novel emerging target list with the validation stage. We see a short list of targets, some trends across those, some challenges and opportunities. And then finally some recommendations. So you can ask multiple questions now to this structured data set about any target ever validated in publications, at least on atopic dermatitis. So if you want to identify novel targets, emerging targets, if you want to assess for a single target what's the evidence across a disease or a group of diseases or a broader therapeutic area to see which indication and target to focus on, that's perfectly possible via this kind of approach that I just showed you. All right, so hope this helps, and let's move on to the next use case. This use case will focus on the question, where can smart additive trial design create advantage? So we'll step away from molecules and focus on how diseases are being measured and studied. So let's look into the example of atopic dermatitis. Let's review the clinical trials on atopic dermatitis, where we see the disease being mentioned in title, summary, condition, or the arms and interventions fields. We found 3,000 clinical trials. Let's look at the last couple of years, 1,500, and we want phase 1, 2, 3, and 4 trials. So 878 clinical trials. Now, we don't want to read them one by one. We want to process all of them. So we can extract insights from all these clinical trials, simply adding the question here. I want to understand the designs, the biomarkers, patient stratification, endpoints for all these drugs. At the I had 20 questions. And then we immediately get questions like, what's the main research approach and design for AD? Is it randomized control and so on? I click on show preview and then download, and then I get this dataset. So here we can see in this sheet all the questions that we had asked, the specific ones, the query we used, and so on. And then in each column, we have an answer to each of those questions. So we can see which biomarkers are being measured. We can open up the filter to see ADA, anti-drug antibodies, and the specific ones. So we can easily go through this list to have an idea of what's being measured. In this way, you get a full overview of all the approaches, all the biomarkers. We could have a key summary of the clinical trial design here. What endpoints, primary, secondary endpoints have been used? What are the drug classes or modalities? So we can go by biologics. We can see monoclonal antibodies, immunomodulators, and so on. So let's say I'm interested in immunomodulator. I filter by those, and now I have 72 of 877 that are about immunomodulators. So I can then see what are the biomarkers, what are the endpoints, what are the intervention arms. So I could look at those where at least there's an active comparator. So now we have 38 of the 870 clinical trials. Any patient stratification criteria, so we can see by age and so on if that's relevant. We can see patient inclusion criteria, what kind of organizations are involved. So we can filter back any biotechs, for example, by just searching here for biotechs, and Pharma for competitive analysis as well. We can see what are the drugs being mentioned and specific dose, if there's an adaptive or innovative trial design, how long was the trial, when is efficacy being measured, at what days, and safety as well. We can see if there's combination therapies, filtered by that. We can see some rationale or a biomarker hypothesis, key differences between the arms, to that understanding what kind of maturity level is the validation or evidence of this trial, what's unique about any of those to design the biomarkers and so on, and what's the key differentiation primary versus secondary objectives. We can always filter by, for example, the phase. So you can say, okay, I'm interested in any phase 2 trials. And then we have 12 of those left with all these criteria. We also, by the way, can filter by the specific conditions, by mesh terms of disease, by drugs, drug mesh terms, etc. Dates, recruiting stage, etc. So a lot of other criteria you could filter by. Now, the interesting thing is you can also upload this document into ChatGPT. So that's what I did. I uploaded the clinical trials document. I said, attached are these trials. I want to identify novel approaches for biomarkers, endpoints, and trial design. And after 4 minutes of thinking and analyzing the data set, here's a quick summary. It also refers to specific cells in the Excel file, so it's always validated. It's not just up in the air and hallucination. You have an executive summary here, endpoint reality check, standards. Then we see what are the most novel approaches for biomarkers. So you can see here different approaches and why it matters. We see the same for endpoints. There is novelty showing up. In trial design, the most due diligence worthy design patterns. So that might give some interesting ideas. And then a short list of 10 trials or concepts to do due diligence on, with a reference to the specific cells. So to give you inspiration to be innovative and in this way have a high impact in how you design your trials. Some trends across those are patterns, challenges, and you can basically now interrogate the full data set to pick up what are the novel biomarkers being mentioned, what are the established ones, etc. So there's many ways you can now interrogate this data set. So the essence of this approach is you find any trials on a certain disease, you extract and analyze each one of those in just a few minutes, less than a minute actually. And then all these insights are being extracted from those clinical trials, where you can then segment them to find the ones of interest, but as well interrogate them, for example, in an LLM like ChatGPT or Copilot. So hope this helps. You can do this for any disease, across diseases, or start from a drug, from a certain modality, etc. So feel free to try it by yourself. This can be helpful for clinical scientists, for people in a preclinical scientific role, in intelligence, innovation, etc. So feel free to share with colleagues that might benefit from this. All right, let's zoom out for a moment. So across all those use cases, KOLs, rising stars, academic collaborators, new startups, technologies, IP, assets, targets, biomarkers, clinical endpoints, trial designs, the pattern is the same. The challenge was never really lack of data. It was about how do you find the signal in the noise and doing it fast enough to matter. So what you've seen today is not about replacing experts or judgments. It's about making sure human time is spent only where the signal is the strongest. So work that used to take days or weeks, like scanning and filtering and puzzling information together and debating, now happens in minutes. And that changes how teams are exploring new opportunities, how early they can move, how confident they can be in their decisions. So they can get a lot more done. And if any of those workflows map to challenges you're facing right now, you don't need to recreate what you just saw. After this session, I'll send you a workflow guidance document with every query, filter, AI prompt that I used today, plus the outputs, so you can apply directly to your own questions. You also get 3 days free access if you don't have access yet, plus 1,000 free AI credits to try it by yourself. If you need more, happy to share options to acquire more credits. And if you want a pressure test or real use case, like for a disease area, a new indication you want to explore, and then find interesting technologies, novel targets, early stage assets or startups, just reach out. I'm happy to think it through with you to find interesting opportunities. Thanks so much for your time and for staying curious about how we find signal faster in increasingly noisy scientific worlds. I hope this helped, and I wish you a great rest of the day.